Bismillahirrahmanirrahim, and welcome back to this next video. And this is the third video in the series of videos on the bioinformatics. In the previous video, I've told you how these bioinformatics it gets started. So the previous video was the part one of the genesis of the bioinformatics. So how this all started. I've told you that in the early days of biochemistry, the biochemists that were mainly focusing on to find a better way to represent proteins in terms of formula that would explain their biological or even the nutritional properties. And then they analyzed that the proteins, they are very huge molecules. They are made up of the amino acids, and these amino acids they are picked from a selection of the 20 amino acids. And then I showed you this whole table which was representing the full name of the amino acids, their 3-letter code, and then their 1-letter code for all of the amino acids that are used in the synthesis of the protein. And I've told you that these full names, these 3-letter codes, and this 1-letter codes for these amino acids, they are actually designated by the International Union of Pure and Applied Chemistry Committee. Then I've told you that the biochemists recognized that a given type of the protein will always contain precisely the same number of the total amino acids and in the same proportion. For example, if you talk about the insulin, so it would always contain, for example, like 30 glycines, 44 alanines, the 5 tyrosines, and so on. They also recognized, these biochemists, that the amino acids they are linked together as a chain, which we call is the primary structure for these proteins, and the true identity of the protein that is derived not only from its composition but also from the precise order of its constituent amino acids. So not only the precise number of the total amino acid is important, but also the precise order of the amino acid that is important for the function, for the proper functioning of a particular protein. And I've told you that the first amino acid sequence of the protein, which was insulin, that was determined in 1951. Now, if you talk about the human insulin, currently, as you all know, that the human insulin is made up of 110 residues, or you can say 110 amino acids. So a human insulin, a functional human insulin, will always contain 110 amino acids, and this is the sequence of the amino acid that will be present for the proper functioning of a human insulin. So the number of the amino acid that will always remain 110, and this will be the sequence that will be required for the proper functioning of an insulin. So if you are manipulating the number of residues or you are manipulating the sequence of the amino acids, that is going to lead to a non-functional insulin. So this is the 110 amino acids represented by their single letter codes. Now more than like 71 years later, because the first protein sequence was determined in 1951, we are now sitting in 2022. So after 71 years, like analyzing protein sequences like these, as I have given you the example of the sequence of the insulin, so analyzing protein sequences that remain a central topic of bioinformatics in all laboratories throughout the world. The reason is very obvious. Proteins are very important for the normal physiological functions of any living organisms. Now, if you talk about a little bit history of the sequence analysis, so as I've told you, the first protein that was sequenced, that was insulin, and that was by A. Sanger. So sequencing of the insulin that actually inaugurated the modern era of the molecular and structural biology, and thus biology got its first fundamental data set, that is the molecular sequences. And of course these molecular sequences, these are the sequences of the amino acids for a particular protein. Now in the early 1960s, non-protein sequences, after the sequencing of the insulin, the biochemists they started sequencing of the other proteins. So the non-protein sequences they got accumulated slowly, but the computers capable of analyzing these protein sequences they hadn't been developed in the early 1960s. So in this pre-computer era, the sequences of these proteins they were assembled, they were analyzed, and they were compared manually by writing them on a piece of paper or tapping them side by side on the laboratory walls, or moving them around for their optical alignment. Now, all of these sequences like this optical alignment, now we call it as the pattern matching, and this pattern matching is very important for determining the sequences of a given protein, the sequences of amino acids for a given protein. So the first computational biologists, then they started to manipulate and analyze molecular sequences, the sequencing of the amino acid as texts, as I have given you the examples of an insulin that they were represented. They started representing them by single letter code in the form of the text. And most of the methods for these analysis that had to be invented from the scratch, because the computers, they were not yet developed, the tools and the software, they were not yet developed, so you have to invent them from the scratch. And in this process, in the developing methods for the sequence analysis, it gave rise to a new area of research, which was the analysis of the protein sequences using computers. That was started. And when you start analyzing the protein sequences using computers, this was the genesis of the bioinformatics. So this is how the field of the bioinformatics started. So in the next video, we will continue the discussion and we will start focusing on further topics of the bioinformatics.